Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And within the cell biology, so far what we have discussed about the cell culture technique, we discussed about the differentiation of the different types of cells, and then within the cell we also discuss about how you can be able to isolate the different organelles utilizing the differential centrifugations or the density gradient centrifugations. Apart from that, we have also discussed about in depth different microscopy technique, whether it is the phase contrast microscopy or the fluorescence microscopy or the electron microscopy. And in detail we have discussed about the different types of protocol as well as the procedure what you have to follow to process the samples for the fluorescence microscopy, phase contrast microscopy, electron microscopy, whether it is the scanning electron microscopy or the transmission electron microscopy. And apart from that, we also discuss some of the crucial experiments. So, now today we are going to discuss about the different topic, and the topic is the flow cytometry. So, what is the use of the flow cytometry? You can imagine a situation where you are actually trying to handle a complicated tissue. For example, if you are handling with the tissue and you want to analyze all the cells, then you are actually looking for a system which can be able to analyze them separately, and you do not want them to be separated simply by the density gradient centrifugation or the differential centrifugations. So, let us discuss that, and we will start with the composition of the different tissues. So, as you can see, I have taken an example of some of the tissues what is present in a human body, whether it is the blood, liver, brain or the heart. And what you can see is that for example, the blood, right, the blood is made up of RBC, WBC. Within the WBC you have the lymphocytes, neutrophils, basophils, eosinophils and the platelets. Apart from that, you also have the serum component. So, that is the agranular component or the acellular component. So, that is we are going to be keeping it separate. And like for example, in the liver you have the hepatocytes, you have the Kupffer cells, and then liver is also having the extensive supply of the blood. So, you also going to have the blood cells. Similarly, in the brain you can have the neurons, astrocytes as well as the glia cells. Apart from that, you also might have some of the fibroblast and all other kinds of macrophages and all other kinds of cells. And then talking about the heart, you can have the cardiomyocytes, you can have the cardiac pacemaker cells, you can have the fibroblast, you can have the smooth muscle cells, and you can have the endothelial cells, which actually are responsible for making the arteries as well as the veins. So, as you can see, all these tissues are very composed of the different types of cells, and you can imagine a situation where you are interested to study these cells, but you do not want to disturb them or you do not want to hurt these cells by going through with the extensive cell centrifugations or some of the purification techniques. So, how you can be able to achieve that? You are looking for a system which actually can separate these cells under the flow, and then you can be able to individually study these cells. So, when you want to study an individual cell, or when you want to distinguish a particular cell from the other cell, you have the 3 to 4 different criteria what you can use. So, as far as the cell is concerned, what you can have the shape. So, different cells will have the different types of shape. Then you can also have the density of that particular cell. So, the number of different types of organelles what is present in a cell is actually going to compose and be responsible for the density of that particular cell. And then you can also have the different types of receptor onto the cell surface, and that also can be a criteria to distinguish one cell from the another cell. And then at the end, the different types of cells may also have the differential metabolic behaviors or metabolic reactions. For example, in some cells you might have some reactions which is responsible for production of the free radicals. For example, in the immune cells you always have these kind of things. Then in some cases you might have the calcium flux, In some cases you might have the ATP productions and all that. So, basic pathway maybe remain the same in different types of cells, but all the other type of cells which is responsible for giving them the functional outcomes may be different in different types of cells. So, all these 4 criteria can be exploited in a flow based assay, where you can actually be able to utilize these parameters to distinguish one cell from another cell. So, if you want to do that, for example, in the mammalian cells you have the different types of sizes, like for example, the sperm cells are having a size of 3.86 nanometer, micrometer. Red blood cells are 5.76, lymphocytes are slightly bigger, 6.29, and what you can see is the macrophages are in the range of 21 micrometer, cardiomyocytes are even bigger than that. So, they will be of 30 micrometer, and the megakaryocytes are very big. So, they are around 40 micrometer. So, what you can see is the size in terms of size, the different mammalian cells are different from each other. Similarly, the cells are different in terms of the density as well as the metabolic reactions as well as the cell surface receptor what is present on these cells. So, these are the 4 criteria one can easily use to exploit, and one can easily use to distinguish the different types of cells. So, for analyzing these cells, you are looking for a system which actually can perform these criteria for an individual cell. So, for that kind of thing, what you are looking for is you are looking for an instrument, or you can be looking for a cell analyzer which could be able to, first you can imagine that you have a cellular populations where you have the cells of the different colors. So, what I am showing is a different color means it is going to cell have a different types of metabolic reactions or different types of cell surface receptors, or it could be of different sizes, like this is a big cell or this is a small cell. So, irrespective of that, first you are looking for a system so that it actually can distinguish and analyze this cell mass and give them a single cell. And once the single cell is coming out from this tubing, it could be illuminated with the help of a laser, and when you illuminate this cell, you can be able to have the detectors. These detectors could detect the size of this particular cell, or you can have the detector which can detect the density of this particular cell, or you can have the detector which actually can detect the fluorescence. So, from where you are going to get the fluorescence? For example, if this cell has the different types of receptors, then what you can do is you can just simply add the antibodies which are actually going to bind to this particular cell, and it is actually going to give you the fluorescence, and that can be detected with these kind of the fluorescence detectors. And then at the end you also require a cell collector. So, in case suppose you want to collect this particular cell and you want to further analyze and perform some experiments, then you can actually have a funnel kind of thing, and that actually can be used to detect or to collect these cells. So, this is a basic instrument or basic infrastructure what you require if you would like to analyze a complicated mixture of cells utilizing the cell analyzers. Let us see how the instrumentation look like. So, in a flow cytometer, the instrumentation is very simple to that. What you have is you have a sample which is actually going to have a mixture of cells. So, once this mixture of cells are going to enter into a tube, it is going to be first focused, and then you are going to have the single cell coming out from this tubing, and then this single cell is going to be illuminated by a laser source. So, once this single cell is going to be illuminated by the laser source, it is going to give the signal, and these signals can be detected by the different types of detectors, whether it is the detector for the forward scanning, or it is the detector for the side scanning, or whether it is a detector for the different types of fluorescence channel, whether it is the FL1, FL2 or the FL3. All these are then going to be processed by the processing unit, and then it is going to be converted into a readable signal, and that actually is going to be analyzed by the computer, and at the end you are going to see the readouts from this particular individual cell. So, if you see this particular instrumentations, what is for the cell cytometer, what you have is you have the 3 component. One is this particular component which is called as the fluidic components or the fluidics. So, that is the fluidics component where you have the fluids, where you have the water, and then this is the component which is called as is a part of the optic system. So, that this is a system which is actually analyzing your sample or the signal what is coming out from your sample, and then this is the signal system what you have required, this is called as the electronic system. So, that electronic system is going to convert this optical signal into a readout signal. So, it is actually going to convert that photon what is coming, what is being perceived by the optical system, and then it is going to convert it into a readable signal, and that is going to be shown by the computer. So, in a typical flow cytometer you have the three components. You have the flow system which is called as the fluidics, and the job of the fluidics is that it is going to focus the sample so that you can be able to have the single cell at a time, and then this single cell can be illuminated with the laser. So, that is a part of the optical system. So, optical system is composed of the lasers on one side and the detectors on the other side. And then the third system is the electronic system or the signal processing system. So, the electronic system is simple, that it is going to convert that signal what is coming from the optical system, and then it is going to convert it into a readable signal. So, let us discuss about all these systems individually. So, in the flow system you are going to have the flow cell, and the flow cell, the purpose of the flow system is that it is actually going to focus the sample so that you are actually going to have the single cell at a time. So, this focusing of the sample is being done with the help of the Schwitt fluid. So, what you have is when the sample is injected into this flow cell, the flow cell is actually going to have the Schwitt fluid. So, you have the outer Schwitt fluid, and that outer sheath fluid actually contains the 0.9% saline or the PBS, and then it is actually enclosing the central core. So, in the central core you are going to have the sample, whereas which is actually going to be surrounded by the sheath fluid, and then it is going to use the hydrodynamic process to focus the sample. So, you can imagine that you have the sheath fluid running into this. So, this is a typical design of a flow cell. So, once the sample is entering into this flow cell, so what will happen is it is actually going to focus this sample, and then it is actually going to give you the individual cells. So, you can imagine that you have the multiple cells at the base of this particular sample, but then ultimately it is going to give you the single cell, and that happens because the sheath fluid is running on top as well as the bottom of this sample, and this sheath fluid, the speed of this sheath fluid is being controlled in such a way that it is actually going to focus the sample, and as a result the single cell is going to come out from this fluidic system, and then this single cell is going to be illuminated by the laser. So, this is actually a typical way in which the sample is going to be focused by the hydrodynamic focusing system, and these fluids are flowing in a single channel, but they do not mix because the speed of these fluids are very different, and the density of the liquid what you use for the sheath fluid versus the density of the liquid what you use for preparing the sample is different. So, that is how they are actually being flow around the sample without having any boundary. So, if you imagine that there is a boundary, there is no boundary. The sample is also running into the liquid, this sheath fluid is also running as a liquid, but because the sheath fluid is running very fast and it is in a focused way. So, because of that the sample is getting focused, and then the single cell comes out from this particular fluidic system. So, once the single cell comes out, then these single cells are entering into the optical region where you have the different types of source light as well as the detector lights. So, once this enter into the optical system has two systems. One is the source through which you are going to use to illuminate the sample. So, you can have the laser as a system, laser as a source, or to the arc lamp. In some cases, the people are also using the argon laser. So that argon laser is actually giving the flexibility to excite the sample at 488 nanometer. The laser is always being used because the laser is giving you the very high intensity and the very high intensity, and that's why the laser is preferred over the some kind of tungsten or arc bulbs. Because the intensity of the bulb is always been varying, whether it is from the beginning or the end. So that fluctuation could actually be problematic for the instruments to, or for the samples to to get excited, and some cases the lamp based excitation is also going to damage the fluorophores. So this is the light source, and as far as the detector is concerned, you have the multiple detector, the detector which is actually going to detect the forward scanning or the forward scattering, or you can have the side scattering. So what is mean by the forward scattering and side scattering is that you can imagine that you have an object, and then if you are illuminating it with a laser beam, so what will happen is once the laser beam is going to hit this sample, it is going to be scattered in two directions. One, it is going to be scattered in a cone shaped structure like in the forward directions, or it is actually going to be scattered in a 90-degree angle. So this is called as the side scattering, which is called as SSC, whereas this is called as the forward scanning or the forward scattering. This forward scattering, as you can see that the forward scattering is depending on to the size of this particular thing. So the FSC is actually going to give you an idea about the size of the particle, whereas the SSC is actually going to give you the idea about the density of the molecule, because as the molecule will, as these laser beams are going to hit the material what is present inside the cell, it is going to be scattered by a right angle. So if the right angle transition, if the material is less dense, then the scattering what you see in the 90-degree is going to be less, whereas the scattering would be in the forward direction is going to be more. But if the material what is present inside the cell is very dense, then the lot of proportion of this laser beam is going to be go into the side scattering, and that's how the side scattering is going to give you the idea about the density of the sample, whereas the forward scattering is going to give you the idea about the size of the bead. So these two channels are being used to detect and to differentiate the different types of cells based on the size as well as the density of the molecule. So the forward scattering is for the size and side scattering is for the density, which means if a molecule is more denser, it is going to give you the more side scattering. If the molecule is of bigger size, then it is actually going to give you the more of forward scattering. And how these two properties can be used? So these two properties can be used even to distinguish the different types of cells. For example, when you are processing a sample, you might actually process the a complicated, for example, if you process the blood, like, so as we see, like the blood is having a size of like RBCs, then you have the macrophages, and then you have the monocytes and all those kind of cells. So what will happen is, if you process the blood, and suppose you are processing the side scattering on this side and the forward scattering on this side, then what will happen is you are going to see the cone shaped dot plot like this, and where all these dot plots are actually going to say that what cell are actually corresponding to which. So this means these cells which are actually going to have the less amount of the side scattering and less amount of the forward scattering is actually corresponding to RBC, because RBCs are of very small size and RBC does not have the nucleus and organelles. So it is its density is also very low. So that's why on the left corner you are going to see the RBCs, whereas on the upper right corner you are going to see the cells which are of very big size, and they also have the very high density. And so what are these cells? These are the monocytes or the lymphocytes, which are being present onto the red corner of this. So that's how you can be able to separate the different types of cells without even going through with the additional detectors. So apart from these, you can also have the additional detectors for the fluorescent channels. For example, you can have the FL1, you can have the FL2, you can have the FL3, and all these FL3 means the fluorescence channel 1, fluorescence channel 2 and fluorescence channel 3, and all these FL1, FL2, FL3 are actually be the dedicated channel for the specific fluorophore. Then you have the electronic system. So, in the electronic system actually converts the photons what you are going to get or what you are going to measure by the optical system into a photoelectron, which means it is actually going to convert that into the electronic current, and that current is going to be further convert into a readable count. So, it actually allows you to measure the amplitude, area and the width of the photoelectron pulse, which means wherever what is the height of this particular signal, what is the area of this particular signal and how many signals you are getting, that is actually going to be the purpose of having a photoelectron systems. If it amplifies the pulse either linearly or logarithmically and then digitizing the amplitude pulse. So, electronic system is only to take the signal from the optical system and then it is actually going to convert that into a readable count, and that is going to be visualized by the person who is doing the experiments. You have the different types of plot what you can use in the flow cytometer. So, you can have the single color histogram. So, what is mean by the histogram is that you can have the single property, for example, you can have the FL1. So, what will happen is you can have the histogram like this, or so for example, this is the unlabeled sample. So, this is the unlabeled sample and then this is the labeled sample. So, you can have the single color histogram. You can have the two color dot plot. So, dot plot means you can have the pattern of the signal. For example, on this side you probably can have the FL3, on this side you can have the FL1, or sometime you dot plot you can have the SSC versus FSC. So, what it is going to show you? It is going to show you the distribution of the different types of signal in this particular plane. So, you can have a distribution. So, what distribution will say that these cells which are present on this side is actually high in FL3, whereas the low in FL1, whereas the cells which is present here is actually high in FL1 and low in FL3. So, that is the purpose of having a two color dot plot. Similarly, you can have the contour plot. So, contour plot is actually an extension of the dot plot. So, a dot plot is actually going to give you the dots, whereas the contour is actually going to give you a contour like covering of that particular signal, and then it is actually going to say that whether the signal is going up or going down. So, the contour is actually going to show you the circularized pattern, whereas in the center it is actually going to show you the highest signal. So, that is how it is actually going to give you the contour signal, and then you can have the two color density plots. That density plot is also like the two color dot plot, and it is also going to show you the density, like it is very simple to similar to the contour plot, but it is actually going to show you the density of that particular sample in that particular region. So, that is how you are actually have the flexibility of different types of plots, whether you have the histogram plot, whether you have the dot plot, whether you have the contour plot, or whether you have the density plot. All these, the purpose of all these plots are to look for the individual properties. For example, you are going to use the histogram plot when you are looking at the single property varying in the different types of cells, but you can have the dot plot when you are looking for the two different properties and how the two different properties are varying in a single cell populations or in a multiple cell populations, but on an individual cell you can be able to ask whether this cell has the two properties or single properties or both properties. So, that is how you actually, if you have the two color dot plot, it actually gives you the more flexibility in analyzing the samples compared to that you have the histogram plot. So, histogram plot always gives you the quantitative informations, or it is actually going to tell you that what is the level of this particular signal, which means on this side if you are putting the FL1, you can have the the fluorescence actually. So, it is actually going to tell you what is the level of this fluorescence compared to this. So, it is actually going to give you the quantitative or the semi-quantitative informations. Also, it is going to tell you the distribution of the samples within this particular histogram plot, but it is not going to give you the information about whether that particular cell is having the second fluorophore or not, because it is only single color histogram, whereas in the case of dot plot it is only telling you the distribution of the two properties in a sample. So, that is actually, so two color dot plot is always being used to qualitatively see how the two properties are distributing within a cell populations, and then based on this you can be able to choose a particular population, and then you ask what is the level of the one signal or another signal. So, we are going to take a few example of this, and then you will understand more in detail. Then you have the data acquisition. So, data acquisition is very simple. You have to process your sample, and then ideally what people do is they are always collecting the 10,000 counts. So, in a flow cytometer you do not call the number of cells, you always call it as a count. So, because every individual cell is considered as a particle, and that particle is being counted. So, what people normally do is they always collect the sample of 10,000 counts. So, when you are going to put the sample into the flow cell, or when you are going to connect the samples to the machine, it is actually first going to allow you to collect the sample. So, what you do is first you collect the samples without even going through with the acquisition process. So, it gives you the option to suck the sample without recording it. So, what will happen is initially what you are going to do is first you see whether the sample is good enough or not. So, that is actually going to tell you have to do it with the help of the SSC versus FSC. So, first thing what you have to do is first you have to collect or check the quality of the sample. So, if you do the SSC versus FSC dot plot or the two color dot plot, so then it is actually going to give you the pattern of the sample. So, suppose this is the pattern. So, you can have the two conditions where in one condition I am getting this kind of pattern, or in the other conditions you are getting a condition like this. So, there is no distribution, or suppose this is like this. So, this is actually a sample which is well prepared, because you are getting a distribution of all the cells in the FSC versus SSC, because now you can be able to see all the cell populations or the cell population present in the sample, whereas in this case your all cells are present onto the left corners, which means either the way you have processed the sample, it is actually destroying all other samples, or it is actually causing the aggregates of these cells. So, this because they are causing the aggregates, the cells are always being present at the lower left corners of the sample, and then this sample is not good enough, because then you cannot do the histogram analysis and ask for any questions, because you have not processed the sample properly. Now, once you have this, then you have the choice that you want to collect the dot plot or whether you want to collect the histogram plot. Then after once you see that, then you can ask that you want to collect the histogram plot. So, then you can do FL1 or FL3 or whatever, and then it is actually going to give you the sample for the FL3. So, FL3 or FL1 you are always collecting in terms of the log scale. So, you can see that it is actually going to give you 10 to power 0, 10 to power 1, 10 to power 2, 10 to power 3. Now, here also you have to always consider that when you are looking at the histogram plot and you are looking for the histogram plot of unlabeled samples, you can have the multiple free, right? Because unlabeled sample is going to have the very low level of fluorescence, it is always going to be like this. But you have the flexibility in a flow cytometer that you can be able to increase the current as well as the gain of the instruments. So, what is mean by the current and the gain is that it is a virtual way of giving the more intensity to the sample. So, when you give the more intensity to the sample, it is eventually going to give you the more fluorescence. So, that is can be modulated with the help of the laser. So, if you provide more current to the laser intensity is going to go up, and as a result it is going to give you the more signal. So, because you can change the current as well as the gain, you can be able to shift these peaks in both the directions. So, for example, if I am using a current of 400, I can just convert it to 700. So, what I have done, I have just increased the current to 1.5 volts. So, in that case the peak is going to be shifted onto this side. If I do the reverse, then the peak is going to be shifted in this direction, because all the thing what you see in the flow cytometer is very relative. It is always relative to what you have done for the control cells. So, in ideal conditions, when you are doing the data acquisitions and when you are trying to do a histogram plot, what you prefer is that you started with 10 to power 0, 10 to power 1, 10 to power 2, 10 to power 3. So, what you prefer is, and on this side you are going to have number of cells. So, what you prefer is for unlabeled samples, it should be around 10 to power 1 and it should give you a peak, so that you can be able to see the change in the fluorescence of this particular labeled sample in both the directions. For example, if I am looking at the untreated versus the treated sample and I do not know whether the fluorescence is going to go up or go down, then in that case, if I keep the peak at 10 to power 1 and suppose the peaks got shifted toward this side when I am treating the sample with some kind of drug or something, and the fluorescence is not going to increase, then in this case it is actually going to give you the peak on this side. And suppose there will be an increase in peak, then it is actually going to give you a peak on this side. Whereas in the other conditions, when you are actually holding the peak of untreated sample very close to 10 to power 0 or less than 10 to power 0, then in that case you will not be able to see any movement of the peak towards the left side, because that time the left side is not existing, because 10 to power 0 is the minimum scale possible in this particular type of analyzers. So, that is why it is important that you keep the peak in such a way so that you could be able to see the movement of the peak on both the sides. That is why the data acquisition has to be done on a multiple occasions, like once you see the control samples and once you see the untreated sample, you have to modulate the untreated sample with the help of the current as well as the gain settings, and then once you got the right positions, then you should not change the current and the gain conditions, and then you analyze your treated samples. And if you see there will be a huge shift, for example, if I see that okay, it is actually crossing 10 to power 3 also, then in that case I can just go back again to the untreated sample, again I will change the current and the gain in such a way so that your treated sample should come within the scale. So, these are the things what you have to keep under the consideration when you are doing the data acquisition. So, first thing, you have to go for the SSC versus FSC pattern and look for whether you are getting a beautiful distribution of the different types of cells or not. It should not be focused onto one region, it should not be focused like here or it should not be focused here, though in both the conditions your sample is not correct, because when you are processing a blood, like for example you are processing a blood, the bloods are having the cells of different sizes. So, it should give you a very nice distributions. Once you see a nice distribution, then you can ask whether the sample has a signal for FL1 or FL2 or FL3 or whatever the fluorophore is present in your cell, and then you do all these modifications with the help of the current and gain, so that when you analyze the treated sample you should be able to get the real picture, because you should have the flexibility, you should provide the flexibility in the system so that the treated sample should move towards the left or to the right. Once you have done the data acquisitions, the gating is also a part of the data acquisition. So, what is mean by the gating is that, see, as I told you, right, gating is the selection of some of the molecules within the whole populations. So, selection of only certain population of cell for analysis of a plot. So, it allows the ability to look at a parameter specific to only that particular subset. For example, as I said, when you start the reactions, first you have to do is you have to do SSC versus FSC signal. Okay. So then what will happen, you are going to get a population. So as I said, these are the populations which is for the neutrophils or macrophages or monocytes, whereas this is the population which is for the RBC. So what gating mean is that if I have collected the 10,000 counts, and suppose I am only interested to see the population of T cell or B cell. So that is not important that you use the SSC versus FSC, you can use any parameter. For example, you can use the FL1 versus FL3 and you can get a parameter. So in a dot plot, what you are supposed to do is you have to gate a particular type of signal. So gate is a electronic way of selecting a particular cell population. For example, I have used the rectangular gate and I have selected this region within this FSC versus SSC, and then what I will do is, so for example, this is a R1 gate. So okay, so then what I can ask is, I can ask the instrument to collect the 10,000 counts, not in the whole cell population. It collect the 10,000 counts only in the R1 gate. So what it will going to do is, it is going to start collecting the 10,000 points or 10,000 counts in this R1 gate, irrespective of how many samples it has collected or how many counts it has collected from the sample. It will not stop until it is not going to have the 10,000 counts in the gate R1. That is actually, and then what I will say is, I will going to say that you are going to show me the histogram, for example, the FL1 for the R1 count or the R1 gate. So it is going to only going to show me the fluorescence of this R1 count. It is not going to give me the fluorescence of the whole cell population, because I am not interested to see the fluorescence of the RBCs. I am not interested to see the fluorescence of the leukocytes or the T cell or B cell. I am only interested to see the fluorescence of the monocytes. So if I want to select the monocytes, I have two choices of selecting the monocytes. Either I go with the SSC versus FSC and I know where the region is corresponding to the monocytes, or I can use the cell specific signaling. For example, I can use the cell specific markers and then I can stain the samples with that particular marker to select what is the region is corresponding to the monocytes, and then I can just do a gating of that particular sample. So I can just make a bracket with the help of the electronic tools what is available within the software, and I can just do a gating for that particular region. So then it is not going to analyze the whole sample, it is only going to give me the flexibility of analyzing the sample what is present within that gate. Gate could be of different types of gates. So it depends on the what kind of shape you prepare. So gate could be a rectangular shape, it could be a elliptical shapes, it could be a polygonal shape, it could be a quadrant gate. So what is quadrant gate is that it is actually going to be a checkerboard kind of thing. So it is actually going to give me a 4 quadrants where you have the lower left quadrants, lower right quadrants, upper right quadrants and the upper left quadrants. So these are the 4 quadrants. So if you have the quadrant gates, it is actually going to give you a checkerboard kind of thing. So that actually is going to, then your whole sample is going to be distributed into these 4 corners or 4 squares, and then it is going to give you the analysis of all these 4 squares separately, and it is going to tell you what is the cell population in the lower left corners, what is the cell population or the pattern of the population in the into the upper left corners and right corners and all that. That is very useful in some of the analysis, and that anyway we are going to see when we are going to discuss some of the experiments related to cytometers. And then you can also have the histogram regions. Apart from that, the gating, you can also do the marking actually. So what is mean by marking is that when you are doing the histogram analysis, what you are going to do is you are actually going to have this, right? Or some of time you can have the even complicated peaks, like you can have like this, okay. So in these kind of cases, what you can do is you can simply do a mark, right? You can have the line like this, you can have the line of the marking. So that is a kind of a gating, but that is only for the histogram plot. So you can just put a marking like this. So then in that case, suppose this is a M1 marker. So in that case, what the machine is going to tell you is, it is going to tell you what is the level of the fluorescence in the M1 marker and what is the cell population in the M2 marker. So you can have the different types of marker. You can have the marker like for this peak, you can have the marker for this peak. So you can have the M1, M2, M3. So you can analyze your sample as per what is present in individual peaks, because now what you see is you are getting the 3 individual peaks in a histogram plot. So if you are interested, you can be able to even measure the individual fluorescence in the individual peak, because that is actually going to tell you whether this the peak in this marker is changing when you are treating the sample with a particular drug or particular molecules or some kind of changes what you are doing, or whether the changes are happening in this. Because if you are looking for the average fluorescence, the average fluorescence of untreated sample versus treated sample may not change, but the pattern within these peaks may change. So that is a very good information what you can extract after even after even collecting the data also. So you can do a marker analysis, and that also is going to tell you some more information about the samples. Then these are the different types of fluorescent dye. You have a huge list of the fluorescent dye what you can use in the cytometers. So what I have shown is only the popular ones like FITC, PE, PERC-CP, APC, Texas Red and all that. What you see is all these fluorescent dyes are doing a emission at a different wavelengths, and that can be used even to label the sample with the different dyes, and that is how you can collect. The problem with the different types of dyes, especially if you are doing the 2 color labeling, is that you have to go through with the process of compensations. What is mean by the compensation is that you know that every dye has 2 different types of spectra. One is called as the excitation spectra, the other one is called as the emission spectra. So in a standard fluorometer you do not have the excitation changes. So what you use is, we always use a 488 nanometer laser. So what you going to do is, you are going to excite everybody with the 488 nanometer. So that is not any problem. What the problem is in the emission spectra. For example, if one dye is having a emission spectra like this, the other dye, suppose this is the FITC, okay, and the other dye is, suppose you are using the phycoerythrin or PE, right, and the PE is having a fluorescence spectra like this, emission spectra like this. Now if I try to see a sample and I am only interested to see the FITC or PE, okay, but what you do not see is the sample what is region of this, right. So these two, if you are using these 2 dyes as a pair, what you see is there is a overlapping region of their emissions. This means the sample is going, the FITC signal will bleed through into the PE signal, and the PE signal is bleed through into the FITC signal, which means there is not going to have any impurity of the sample. Ideally what I should have the signal like this. I should have a fluorescence signal of FITC like this and the PE for this. In that case, this is the ideal condition where there will be no overlap of these 2 dyes. So whatever I see is actually a pure cell. So if I see a signal which is only having the FITC signal, it is actually a pure signal. But in this kind of situation where you have the overlap of the 2 different types of dyes emissions, this is the region which is actually going to create trouble. So what I have to do is, I have to collect the sample and I have to cut down the sections. I have to cut down the data in such a way so that I should, you know, if I suppose this is a background, right. So this is of 0 fluorescence, right. So if I increase the level of my background and if I make it slightly bigger, for example, if I go up to this, then I am achieving this particular kind of conditions. But what is happening when you are going through this process of cutting the data, you are actually losing the data. For example, you are losing this much from the FITC and you are losing this much from the PE, which means these are the things what you have to do when you are using the two different dyes which are having the overlapping emission spectra, and that is the process is called as the compensations. So, when you use the two dyes, the software is going to do the analysis and it is going to tell you that how much is the background signal you have to uplift, so that you will be able to overcome the compensations, or in some cases it is actually going to do the integration of these two signals, and it will tell you that if you see a FITC signal, it is going to be contaminated with the 15% of the PE signal. If you see a PE signal, it is going to be contaminated with the 25% or 22% of the FITC signal. So, that is a kind of a subtraction it has going to put when you are going to go through with the analysis process, and that is how the compensation is going to help you to overcome this kind of situations, and that it's always been inbuilt into the software. It is going to allow you to go with the compensation process. Now, let us discuss about the different types of experiment what you can do with the help of the flow cytometry. So, in the flow cytometry you can do the apoptosis, cell cycle, DNA content, Ross generations, phagocytosis, oxygen burst and the cytokine array. Ideally, the flow cytometry can be used for 2 or 3 different types of experiment. What kind of experiment you can do? You can do an experiment where there will be a change in shape or size. Okay. So, if there will be any change in shape or size, you can use the flow cytometer, because it is having a FSC versus SSC module. So, that can be used to detect whether there will be a change in shape and size. Number 2, you can actually do fluorescence or cell cytometer if there will be a appearance or the disappearance of a antigen or the molecule from the cell surface, because then you can do imaging, or then you can do the staining of that particular sample. Number 3, you can also use the cytometer when there will be a change in the cellular content as well. So, for example, you can, or there will be appearance of a particular object. So, these are the 3 different criteria which are mainly being people use to design the experiment or to consider the cytometer as a potential to answer the questions. These are the examples what we are going to discuss, but these are not the only thing what we can do with the help of the flow cytometer. We can do even further up. So, as far as the apoptosis is concerned, apoptosis is a process which is also called as the programmed cell death, and the programmed cell death is always been resulted into the degradation of the DNA as well as the externalization of the phosphatidylserine onto the cell surface. This means, this is the kind of examples where you are actually going to see a change inside the cell as well as the change outside the cell. So, what you can do is you can actually stain the DNA with a DNA dye. For example, you can use the propydium iodide, or you can stain the some reagents, so which actually going to bind to the phosphatidylserine. So, for example, you can use the annexine. So, annexine 5, which is actually labeled with FITC. So, you are actually can use the annexine 5, which is labeled with FITC. Annexine 5 has a very high specificity for the phosphatidylserine, which is being present onto the external surface of the cell surface, and you can also use the propydium iodide. So, propydium iodide always gives the red signal, the FITC is going to give you the green signal. So, if you stain the cells with the red the PI as well as the green signal, the PI is going to label the dead cells, or whereas the FITC is going to, or the annexine is going to label the apoptotic cells, and that is how you can be able to analyze. Apart from this, you can also use the another combination which is also called as the cell combination like acridine orange versus the PI, so propydium iodide. So, if you use these two dye as well, you can still be able to detect the signal, because what happen is the acridine orange is a dye which enters into the cell, because you know apart from there will be externalization of PS onto the cell surface, the cell membrane permeability is also being compromised in the case of apoptosis. So, if there will be any compromised cell permeability, it is going to allow the movement of these dye. So, acridine orange is a specific dye which always goes inside the cell and it going to label the DNA only if the cell is compromised and the cell membrane is going to be compromised, whereas the PI is going to label the cells irrespective of this. So, this is the data what you see. So, acridine orange and PI if you stain, and this is what I was talking about, the checkerboard analysis. So, what you do is when you stain the cells, you are going to have the two samples, one is untreated sample, the other one is treated samples. So, then what you do is you just do a checkerboard analysis. So, these are the healthy cells. So, what you do is you do a checkerboard analysis and only cover the healthy cells, and then you are going to analyze the treated sample. So, what you see is that the upper left corner, upper right corner and the lower left corner and the lower right corner. Now, these are the cells which are actually going to have only the propydium iodide. So, these are the cells which are actually the dead cells. These are the cells which are having the both the dye in a high concentration. So, these are the apoptotic cells, these are the also apoptotic cells, and these are the healthy cells. So, that is how you can be able to use the staining techniques with the different types of dyes, and you can use the checkerboard analysis with the help of the gatings and that is actually going to tell you what is the cell population within the sample is apoptotic. Then we can talk about the cell cycle. So, you know that the cell is going through a different cell cycle, and that is actually going to happen that it starts from the M. So, in the M, then it is going through a preparative stage, it is making a G1, then it is entering into the S phase. At this stage, the DNA is going to be double and then it is entering into the G2. So, DNA is still double, and then at this stage the DNA is going to be cell is going to be divide, and that is how DNA is going to be normal. So, you can imagine that if I am starting with the n amount of DNA, it is going to be 2n at this moment, and then again it reaches to the n. So, if I want to study the cell cycle, what I can do is I can just stain the cells with the propydium iodide and I look for the content of DNA. Okay. So, ideally what will happen? Ideally in the G1 you might have seen that it is actually the n, G2 also it is 2n, and the S it is actually 2n. So, that is what happened in the this is the your G1 phase, this is your G2 phase, and the in between what you see is actually the S phase. So, once you characterize the G1, S and M, the G2 phase, it is actually going to give you, it is going to allow you to study the cell cycle. So, what you can see now here, this is the untreated sample, this is the treated sample, and what you see is there will be a disappearance of the G2M phase, which is actually this phase, and there is a increase in the G1 phase, which means the cells are actually not going beyond the G1 phase, which means they are not entering into the S phase. Then you have the phagocytosis assay. So, you know the phagocytosis where if you add a particle to the macrophages or any phagocytic cells, they actually engulf that particles. So, in this particular assay what people are doing is they are just simply using the RBCs and they are labeling it with antibodies, and the basic principle is that you can actually label these RBCs with a dye which is called as the FITC. So, what happen is when these FITCs are present outside, you can have two conditions where the particle is present outside or particle is present inside. So, when you are actually having a particle outside, it is very sensitive for the trypan blue, because the trypan blue is a very good quencher for the FITC. So, it is not going to allow the FITC to give you the signal, and that is how you can be able to discriminate what is the amount of the particle present outside and what is the particle present inside, because you know that trypan blue will not enter into the healthy cells. That is why this signal is going to be remain intact, whereas this signal is going to be quenched. So, this is what exactly going to happen. These are the normal cells. As I said, you always have to take the healthy cells first, and then these are the untreated cells, and these are the treated cells. So, what you see is that this is the cell population which is actually has taken up the trypan blue, and that is how it is actually showing you the fluorescence signal. Then we have the cytokine array. So, because the the FACS or the flow cytometer is has the potential of analyzing the different sizes of the objects, as well as it is actually having the flexibility of analyzing the different types of fluorescence. The people have also designed the cytokine arrays, which actually can allow you to measure these different cytokines in a different in a particular given sample. So, what you people are doing is they are taking the beads of different sizes as well as the different fluorophore. So, these beads are actually having the different fluorophores. Mostly these beads are the quantum dots what they are using of different sizes. So, you can have the 1 nanometer, 10 nanometer, 100 nanometer. So, all those kind of sizes of the quantum dots which are actually going to give you the different types of fluorescence. And now, what you are going to do is you tag these dyes or the beads with the antibody, and these antibodies are going to be specific for a particular type of cytokines. Now, what you are going to do is you are going to develop all these beads and then you mix them. Okay. So, you have the different sizes beads, you have the different fluorophore beads, and then all these different sizes as well as the different fluorophore containing beads are tagged with the different antibodies. So, you can have the antibodies for TNF alpha, you can have the antibodies for IL-1 and so on. So, now what you are going to do is you analyze, you incubate these beads along with the cell supernatant or the cytosol where you are interested to measure the cytokines. So, what will happen? These antibodies are going to bind their respective cytokines, and now what you are going to do is you are going to analyze this into a FACS machine. So, what the FACS is going to do is it is going to separate these cells based on the size as well as based on the fluorescence. Now, what will happen is that if they are actually going to have the different types of binding. So, once you added it, you can have the separation based on the size as well as based on the fluorescence, and height of these beads are actually going to tell you the concentration of that particular cytokine present in the cell supernatant. So, as you can see here, this is the one bead which is actually going to give you a histogram, and that is corresponding to the IL-10. Similarly, you have the beads which are corresponding to IL-17, then these are the beads which are for TNF alpha. So, because they are actually making the individual histograms, you can be able to very precisely say what is the level of TNF alpha, what is the level of IL-16 and all that. So, you can be actually having the flexibility of measuring all the cytokines in a given sample in a single shot, both for the control as well as for the treated samples. So, this is all about the flow cytometer and how you can, what are the different components are present in the flow cytometer, and how you can be able to modulate the conditions so that you can be able to analyze your samples. And with this, I would like to conclude my lecture here. Thank you.